Okay, welcome back. So this part, do you have these slides? Or let's see how it goes. So this next hour will be tech team. I'll do the first half, and Professor Shukla will do the second half. So let's see how it goes. So I mentioned earlier that the 5 enzymes. So this is the last bit of biotin-dependent enzyme. So the last bit is the PC, pyruvate carboxylase. So you use pyruvate as the substrate. And as a reminder, PC is right here, converting pyruvate to oxaloacetate. Also, it's involved in basically gluconeogenesis and many other processes. Okay. All right. So now let me change to. Yeah. Do you have this slide? Yes. You do? Okay. Good. All right. So I can talk more about PC other ways, but for today, only focus on the reason I want to bring it up is this small molecule, saccharide-AMP. Have you heard of it? Saccharide-GMP. Have you heard of that? Yes. Saccharide-AMP? Oh. All right. Old news to some of you, but I guess others may or may not know. So let me just go through that briefly. So it was discovered 2008. It's a broadly considered second messenger for many bacteria, for bacterial growth and infection. And so saccharide-AMP, 2 AMP molecules forming a cycle, 3', 5', essentially. So important cellular functions, and also saccharide-AMP secreted bacteria can also cause host immune response, this and that. So at that time, when we started working on this, a lot is known for RNA molecules, aptamers and stuff binding riboswitches, binding saccharide-AMP, but how it regulates proteins was not known. So Ron Breaker actually studies these riboswitches quite a bit, but this is a collaboration with another group, who actually worked on listeria, so a different microorganism. So it's one of the most virulent foodborne pathogens. So in the US, probably in India as well, we often hear recalls of food because it's contaminated by listeria. For example, collaboration with Joshua Woodward, or Josh, at the University of Washington in Seattle. So he's a microbiologist working on listeria, looking at saccharide-AMP signaling, essentially. For example, ice cream can be recalled, and whatever, that's okay. So on this slide, I guess you don't have, so I forgot to put in, my apologies. But the idea is, I guess, to explain how he found the target. So using saccharide-AMP. So using chemical proteomics approach to identify targets of saccharide-AMP in this microorganism. Okay. So essentially, he made a column that contains saccharide-AMP. And then he put lysate or listeria onto that column, and then use free column as a control. And then after that, omics. So from this, he identified many, many targets like this. And I should say, we had a good collaboration with Josh. And many of these you can see are hypothetical, in the sense that, which means they're not really very well understood. So, but then this was 2014 when the paper was published. And then since then, we have worked on many of these. We got the crystal structures of them, and then Josh did some functional work. So many of these actually published. There's functional understanding what the effects are. But then down the list here is PC, pyruvate carboxylase from listeria, which is up here. One of the good, strongly pulled down by the column, by the Bs that contain sarcodine AMP. So because this one has a known function, pyruvate carboxylase, so he sort of got drawn to this target. And then again, he did a literature search, found out I'm the expert on biotin-dependent enzymes. So he reached out to me and said, okay, can we get the structure of the stereo enzyme bound to sarcodine AMP? So that's how I started. Okay, so PC. So I already showed you a little bit. It's a crucial enzyme for gluconeogenesis, lipogenesis, and replenishes TC cycle intermediates. I showed you that as well. It's a tetramer, so half a megadalton. But all of this work was done by crystallography before cryo-EM. Okay, before we learn how to do cryo-EM anyway. And it's stimulated by acetyl-CoA. And then, as I mentioned earlier already, PC deficiency is linked to diseases. So disease-causing mutations that are basic children born with this mutation will have this problem, lactic acidemia, hypoglycemia, all of these very serious problems with PC deficiency. So this is the gluconeogenesis pathway going from pyruvate up to essentially this is the compound that leads to glucose. So what Josh did is using purified Listeria enzyme, I think we purified, sent it to him. He has a binding assay. And then so here's the binding curve. And then based on that, you can get the KD, so about 8 micromolar or so, roughly half height of this. And then he also did enzymatic assays. Well, you see that again. So in this case, you can see the compound binds, the Vmax goes down, but the Km doesn't change, as shown right here. Km is a light symbol here. Km is pretty much bounce around, but doesn't really change so much. Vmax, as you can see, when you have more and more sarcodine AMP, it went down a lot. So that means, again, it's not competitive, because it does not, if it's competitive, when you have more and more substrate, it should compete out the inhibitor. So very likely, it's also allosteric. How it works, we didn't know at the time, so this is sort of why we collaborate with them. In addition, it seems like this inhibition is sarcodine AMP selective. So he treated the enzyme with sarcodine GMP, really no inhibition. Similarly, with the CGAMP or sarcodine AMP or PAPA, very weak activity. So sarcodine AMP seems to be very active against this enzyme as an inhibitor. So we determine the structure of this 500 kilodalton tetramer by crystallography, and then sarcodine AMP bound right here. But before I go there, so remember BCCT? BC is a dimer right here. The other dimer right here, tetramer, so 4 BCs. So the whole structure is like a square or a diamond. And then each corner is a dimer. BC dimer at one corner, opposing corner, another BC dimer, CT dimer, CT dimer. CT is a tin barrel. I heard about tin barrel yesterday. So CT actually is a tin barrel with additional flourishes on the top, whatever. And then BCCP in the middle, right here. So BC, CT, BCCP, and then another domain which I won't talk about too much. And then sarcodine AMP actually binds at the dimer interface of CT. The molecule you can see is twofold symmetric. So it binds right on the twofold axis of CT. But still, it's quite far from the active site. The active site is signified by this metal ion in the CT active site. So definitely it's allosteric. So then the question is, how does binding a compound way out here control catalysis? So first you look at how it bound. So here, sarcodine AMP, 2 adenines are stacked against 2 tyrosines. And then the phosphate recognized by the serine through water, and then a tyrosine at the bottom. And then this serine on the other side as well. So essentially, you can look at the binding side. You have the 2 tyrosines stacking against the 2 adenines, and then the phosphate right here. And then you have actual stuff in this binding site. So that suggests that this binding site probably can accommodate other things besides sarcodine AMP. What that is, we don't really know. probably have to do the reverse now. You can use PC as a bait and do a pull down, do metabolomics. Nowadays, I guess you could do that and then see what come down with it. Maybe cyclic AMP will come down for sure, but probably something else will come down, which will fill these two pockets as well. Well, I have no idea what that is. So, and then I should also mention the second thing I have no idea what that is also. As I showed you, cyclic GMP doesn't work. But if you look at the binding mode, adenine, pi stack against tyrosine, G can, I think, should be able to do the same thing. So then why is cyclic GMP cannot bind and inhibit the enzyme? We still don't really understand. So the enzyme, the listeria enzyme is inhibited by cyclic AMP. The human enzyme is not. So the question is, how does that work? So if you look at it, the tyrosine that stack against the adenine is a threonine in human enzyme. So the pi stacking is gone. And then you can also see there are other changes in other residues which are highlighted red here. I think you have that slide, I think. So you can see many differences between the different species. So the E. faecalis has a tyrosine, but then the two alanines are changed as well. The serines changed to a lysine. So what Josh did is he mutated these residues. So the wild-type enzyme is sensitive to cyclic AMP, and he changed the tyrosine to a phenylalanine. It's okay. So pi stacking is important. The hydroxyl is not. Well, the structure says hydroxyl is not really directly involved, so that's okay. And then when he changed the tyrosine to a threonine, which is the green thing, green symbol here, you can see you lost sensitivity. So confirming that the human enzyme is not inhibited, also showing the importance of the pi-pi stacking in the interaction. So how does it work? So when we compare the structure of the, I know this is very complicated, that's what you have. I have a movie to show you in a little bit. When you compare the structure of the free enzyme, which is in gray, and the complex, which is in colored here, you can see dramatic changes. They don't overlap really that well at all. And then if you look at the binding site, in the free enzyme, the two tyrosine actually encroach into the binding site. So they're actually going to clash into the adenine bases. So there are a lot of complementary changes in the binding site as well between the free protein and the ligand bound form of the enzyme. So this is basically what I'm showing you. So here is the free enzyme, and here is the complex. You can see the dramatic complementary changes in the tetramer going between the two states. But then the question is, why is this state inactive? Or why is the bound state inactive? So that's shown on this slide, which you have as well. So you have essentially, in the complex, the four monomers of the enzyme are essentially identical structurally. You overlay the four monomers, they are very similar to each other. The binding site is down here. So overlay is based on CT domain. BC domain can go anywhere, and it's only show 1 degree change difference between the four monomers. If you do the same thing on the free enzyme, overlay on the CT part, everything overlays quite nicely. BC is all over the place. So the enzyme is very dynamic in the free enzyme state, and the CDA stock of the AMP bound form is very rigid. And that's shown in the little movie right here as well. So you can sort of see the difference right here. I didn't morph it, but you can spin it around. You can sort of see the difference in the BC part. So the idea from this is essentially we feel that cyclic AMP maybe have frozen the enzyme into a single state, and the enzyme need to be dynamic during catalysis. So that's how it inhibits hypothesis. Maybe nowadays we can do MD simulations on such a large system to really understand how it goes. So the basic idea is the enzyme need to go through different control states during catalysis, and binding of cyclic AMP make the enzyme stay in a single state. That's why it cannot go back and forth anymore, and it cannot do catalysis. So that's the idea. And then Josh actually went further, essentially, to show how cyclic AMP is actually toxic. Okay, so in listeria, it doesn't have a full TCA cycle. So you start from glucose, it goes down to making amino acid, alanine, aspartate, glutamate, and then PC is right here. So glucose to pyruvate, pyruvate to oxalic acid through PC, and oxalic acid to citrate synthase, which I talked about earlier, and then that gets going further down, give you glutamate. And then in listeria, cyclic AMP is made by an enzyme called diadenyl cyclase. So he made a conditional knockout or deletion of this diadenyl cyclase because essentially it's essential. Oh, essentially it's an essential gene. Sorry about that. So you can see that essentially when you knock out the cyclic AMP, you have a lot of glutamate being formed. So essentially, so cyclic AMP inhibits PC. So when you remove it, it goes down the pathway. You can make a lot of glutamate. And he also showed that the growth in macrophages, because listeria can infect and grow in macrophages intracellularly. It's an intracellular pathogen. And then wild type grow very nicely. When you reduce the levels of cyclic AMP by knocking out the cyclase to biosynthetic enzyme, you don't grow anymore. But then that can be reconstituted by putting in essentially, or by removing the citrate synthase, by removing that. Now you can grow again, but then if you add back the citrate synthase gene on the plasmid, you're dead again. So that confirmed the mechanism that this is whole depending on the entire pathway. And then he also looked at the pathway as I mentioned earlier. And finally, the disease is called through pyroptosis, essentially, by looking at wild type relative pyroptosis. When you reduce the level of cyclic AMP, you can see pyroptosis went up a lot. So that's how the cells die through this process. So cyclic AMP, in the case of citrate synthase, had a tremendous impact on cell growth. And then disabling pyroptosis can restore the cell growth by getting rid of caspase-1. So this is actually a very nice story. It's the first example how cyclic AMP, dinucleotide second messenger, regulates a protein. In this case, happened to be a metabolic enzyme. But as I showed you, based on the omics, the pull-down experiment, he identified many other ligands. Many other proteins that bind cyclic AMP. So there are many other functions in the cells as well besides this one. But this has actually turned out to be a very nice story, and that's why actually it's a quite interesting addition to pyruvate carboxylase. I can say a lot more about pyruvate carboxylase structures, this and that, but I guess you're interested in small molecules, I think. So this is a bigger small molecule. Okay. All right, so that's basically my part of the next hour. So I finished 2 minutes early. So I guess we need to get Professor Shukla here so he can take over his part and then finish up the rest of the story on metabolic enzymes. Well, while we wait for him, any questions? Any other things you want to discuss? I know he said there are some questions you asked him yesterday that he said ask me instead. So I'm here now, if you remember. No? Okay. Should someone go get them? Should someone tell them that I'm done? Could you? Yeah, thank you. No questions. Good? Yeah, go ahead. I'm sorry.
You showed these graphical results now. So what experiment you have done to get these results?
Oh, what experiments? So this is done mostly by Josh in his lab. So he worked with listeria and also using macrophages. So these are using essentially cells. And then infect the cells with listeria, and then different strains of listeria that he used to control, to see how the effects are. Does that make sense? So I showed you the different. Oh, that's lost. Okay. Yeah. So these different strains here. So he has listeria controlling different process. So this is the one that gets rid of the cyclase that produces sarcodiamp. So that strain has a lower level of sarcodiamp. So all of these essentially to see the effect on cell death and also infection.